Mary Power comes to us from the University of California at Berkeley, where she is a professor in the Department of Integrative Biology. Today's lecture marks a homecoming of sorts because Mary is a local girl done good, having grown up in Winchester and elsewhere in the Boston area, and then attending Brown University and Boston University for a master's before heading off to the University of Washington for her PhD. Now, Mary's CV reads in many respects like a history of the field of ecology, at least over the last several decades, as she has been a pioneer in moving the field forward. Back when she was first doing her doctoral work, when interspecific competition was thought to be the primary factor shaping communities, Mary focused on predation. And as you probably know, the role of predation has been increasingly recognized, and her work was among the first to really emphasize its importance. From there, she then shifted to a focus on food webs and the interactions among species at several trophic levels, which again has become a major focus. And more recently, she is looking at the role that the physical environment plays in regulating communities at both the local and regional scale. And I think we'll hear a lot about that today. But it is really true to say that when you look at Mary's record, she has been at the forefront really throughout her career and has really had a very, very important influence. For this reason, she's received a variety of honors. She is a recipient of a Guggenheim Fellowship, as well as the Kempe Award for Distinguished Ecologists and the G. Evelyn Hutchison Medal. Also, she was recently elected to the American Academy of Arts and Sciences. Moreover, Mary has played a leadership role in the field of ecology as it interfaces with society at large. She has been a president in what are arguably the two most important scientific societies in the field of ecology, the American Society of Naturalists, and more recently, the Ecological Society of America, for which she is currently the president. So it's a great honor to have Mary joining us here today. She will speak to us on food webs in river networks, algal-mediated linkages of rivers to watershed, and this is hard to say, and near-shore marine ecosystems. Mary, thank you very much for coming here.
Thanks a lot. Thank you. It's been great fun to be here and just had wonderful, very fascinating conversations throughout the day. It's a wonderful center, many amazing people here, so keep up the good work. And I also wanted to comment on Bank of America. Bank of America is doing a very good thing in the neck of the woods that I'll spend most of my talk on in the California North Coast. They've funded a very courageous loan for some bankrupt timber mills to buy up 55,000 acres of cutover forest and try to restore it as a sustainable redwood forest. It's got great climate, but was really left in bad shape by the commercial companies. But this will be a community-based forest that feeds local communities, and Bank of America has given them a 20-year interest-free loan that made that possible. So it's very interesting to think as we look forward to a more sustainable future how these long distance and sometimes commercial interests can actually be made to support rather than undermine the values that probably most of us share. So it's been great to be here, and I also wanted to comment on food web ecology and a little pearl that I heard that came from Harvard from my first advisor, Tom Shaner, who was a student of Ernst Williams. So Ernst Williams, most of you probably know, is a very famous herpetologist that influenced the school that Jonathan is a stellar representative of, the people who've learned so much about evolution, biogeography, and ecology from the study of Caribbean animals. Am I not? Oh, yes, it should be turned on. Okay, thanks. So Ernst Williams had, I've heard, this law called the law of unsympathetic magic of nature. And that law states that once you come to a firm, vivid realization about how nature works, your next observation will refute it. And there's nowhere that this law is more manifest in ecology than in food webs, because imagine we have this complex web with all these idiosyncratic players that are being perturbed by various environmental complex factors, and we want to find the strong threads that tell me whether this heron will make a difference to whether the world is green or barren or not. Or is there a strong web that links heron through tadpoles, through fish, that will influence something that I care about in the ecosystem? And you might care about the really gross ecosystem properties like green, barren, nitrogen storage, albedo, or whether a particular species will stay on the planet or not. But whatever you want to predict, we food web ecologists, we experimentalists, and there are many here in the audience I've met today, work really hard, do these laborious field manipulations to see what are the rules, what are the strong interactions, which can you discount as weak, and then you repeat those experiments, and in a different context, temporal, how long has it been since the last flood, or spatial, where am I in the watershed, and you often get a different answer. So food web ecology, community ecology in particular, has suffered from this lack of being able to predict. And I think, in part, it's because we've taken too little specific note of what is the temporal and spatial context in which you're learning this. So I think what we need to do to redress this problem is to take context dependency on head-on as the main focus of our study. So I just want to show you how we're approaching that, focusing on algae. The first part of the talk, I'll give you a quick vignette to show why we should all care about algae, although I'm sure in this enlightened audience, you probably all really do care about algae and diatoms and cyanobacteria. But people in general care when we have problems with algae. There are too many algae contaminating our freshwaters, eutrophication, or here's Harvard, so you can locate the eastern seaboard in the sea of green chlorophyll that's excessive from a Nimbus satellite photograph. There's a lot of near-shore problems with too much phytoplankton, too much algae. And a lot of it's our fault, of course, with our over-enrichment of watersheds and coastal oceans. But we should also care about algae when algae is very inconspicuous, because it's still the base of a food web. It's just the base of a food web that we consider high quality here in this mountain headwater of the Merced River. This is a food web where if you wandered into this pond, the rocks wouldn't even feel slippery. You'd have to look under quite a few rocks to find bugs. Not many bugs, but it is supporting predators, not only trout and salmon, but osprey, bear, otter. So if we have water quality situations like this, we often have algae that are cranking away, maybe one division a day, growing really fast and turning out highly edible, highly nutritious cells. And those sustain enough invertebrate production that they can sustain predators, and you can build up the classic inverted pyramid of trophic level biomass, where there actually might be the most biomass might be, in certain cases, in fish. And then the fish suppress these lower trophic levels so they don't become nuisances. They prey on them, or they store nutrients and slow primary production. So those are kind of ecosystem services that we get from this kind of food web that are driven by the food quality of algae. So that matters. So here's a quick vignette about algae. It's going to take place in a prairie stream in south central Oklahoma, just to show you that it's not too hard experimentally to find out that food chains are often implicated in how much algae build up and whether the world looks green or barren. This is a little prairie margin stream, Briar Creek, at low flow, with the pools separated from each other by long, shallow riffles. Here's the pool head, here's a long, shallow riffle, and there's the next pool upstream. And walking down Briar Creek, I was very lucky to take this walk by myself just after I'd read Jim Estey's papers on sea urchins that are wiped out by sea otters, which maintain kelp forests. The sea otters are protecting the kelp forest. So three-level trophic cascade was something was in my head. And see these green pools and then similar pools adjacent are barren. So go snorkeling in the green pools, and sure enough, there are the bass predators. And in the barren pools, those bass are absent, and there are schools of this soft, thin, very vulnerable minnow, Camposima, which is a voracious algivore if it's not eaten by bass. So it sure looked like a three-level trophic cascade to me, and it was quite easy, actually, in this situation, to do the experiments to show that. We simply fished bass out of a green pool, added Camposima to one side, turned it into a barren pool within 4 or 5 weeks, and then we added free-swimming bass to barren Camposima pools and turned them into green pass pools as the Camposima either fled or got eaten. Again, fast dynamics, 4 or 5 weeks. So algae are really good for seeing these trophic cascades. They respond quickly. They don't have roots doing hidden things underground. And weather also rearranges these players, and you can see the consequences. When storms rearrange bass and Camposima, the dynamics also turn them green or barren correspondingly, again, within 4 or 5 weeks. Those storms can happen anytime during the year in this continental climate regime. which is quite different from the place I'll spend the rest of the talk describing, and that's a Mediterranean climate regime on the California coast. So this is where I've worked now 20 years on a tributary of the Eel River. Here's the eel at low summer flow. In winter, this whole channel can fill with water, and a lot of the gravels get shoved out to the Pacific. Eel has one of the highest disturbance or gravel yields of any river of its size in the world because it's cutting through steep, uplifted, but very soft geologic topography. So you take a fragile geology, tectonically uplift it, dissect it with a river, and you've got a lot of fragile land. And so we work in a tributary of the Eel, this South Fork here. Here it is going into the ocean, which has been 8,000 acres of old-growth conifer and redwood forest has been protected by the UC Natural Reserve System, a priceless treasure now threatened by the collapsed California economy. So wish us luck. So here's the seasonality of algae after we get one of those big bed-scouring floods. The water is starting to clear and warm up in March, but the substrate's barren. This is silt on the rocks that's starting to drop out. This is a 2-foot-long steelhead, and these are the tracks of bear that are chasing them at that time. But not much rock-bound life, not much algae, not many insects. Now, fast forward to June, and after these big floods, we tend to see really long growths of this filamentous green alga, Cladophora. It grows, I think of it as Rapunzel's hair. It grows meters long branches and establishes vegetatively from the boulders and the bedrock that didn't move and scour the previous winter. That stuff senesces and it accumulates as floating mats. This is a particularly big algal bloom year. The slack water, these floating mats occur, they're hot spots of insect emergence. The turfs are still attached in the main stem where the water is flowing, but both the mats and the turfs wither back, and there's kind of a stringy knotted remnant you see here in August, and that architecture that's the collapse of the algae long before the next winter floods is driven in part by this midge. But it wasn't obvious just from inspection whether the midge was producing this algal collapse or just entrained to it. Its numbers increase as the algal blooms, especially those floating mats, and then its numbers drop, the midges drop off as the algae collapses. But is it driving the collapse or just tracking it? So that's the sort of thing you can't know without experiments, unfortunately. And so for now quite a few years, we've done these simple experiments of enclosing about 6 square meters of the river and removing fish, stocking them with certain combinations of the rather non-diverse fish there, the juvenile steelhead or a common roach, a common minnow, the California roach. So stock those enclosures, leave some as fish-free exclosures, and get a surprise, actually, that, again, fish play a very powerful role in algal dynamics. They're linked to algae trophically, and they seem to be very bad for algae. So here is a photograph of what was representative of 6 of the 6 fish enclosures the first year I did these experiments, which is the algae started out 60 to 80 centimeters long, and in 5 weeks had collapsed, so there were turfs nowhere more than 1 centimeter high. Here's a 9 centimeter yogurt container core scar, and here's a 11 centimeter high sediment trap and a 8 centimeter fish for scale. So that's in the presence of fish, and in their absence, the algae stayed erect, but it got overgrown with both globular green and brown colonies that I'll describe in a minute, and also epiphytes that turned it from that nice bright green to a scuzzy yellow color. So it's staying erect, but it's changing. Just some data to show you very quickly. These are the unstocked enclosures, exclosures, and non-destructive height measurements and destructive biomass sampling, more algae in the absence of fish. And more of this floating globular stuff, which turns out to be colonies of the cyanobacterium nostoc, 3 orders of magnitude more of that in the absence of fish than in their presence. So why are fish so bad for algae when they're predators? These fish are more, the steelhead's a pure predator, and the roach, large roach, are mostly predaceous. So it turns out it's mediated through that midge I show you. The midge is one of the most common macroinvertebrate grazers most years, not every year, but most years. And I thought fish ate midges, because they certainly do when you take them out of the tuft, tie them in thread, and I've done midges in bondage experiments to see where predation risk is higher and lower in the stream at large. They love midges in that display, but they don't eat them. They don't seem to recognize them as prey when they're in the tufts. But there are small predators that do. These highly visual damselfly nymphs and also small fish fry prevent midges from settling. And so when you eliminate the fish in exclosures, you get these small predators recruiting in large numbers, and they can wipe out the midges. So that's what's going on. And some nested experiments confirm that that was the mechanism that I won't go into here. Basically, though, that first experiment was a whopping, if you have fish, larger fish in the system, you have a 4-level system in which they suppress these small predators. What's in the guts of these large fish is mostly herbivorous insects. So if you just did diet studies, you'd think that they would be good for algae, eliminating the herbivores, because they mostly eat mayflies and herbivorous free-living midges. But there's one herbivore they can't get, and if these small predators are eliminated, then this guy is released, and that's sufficient to mow down the algae. So here's diet doesn't predict dynamics, yet another example of the inconvenient truth that you often have to manipulate, or at least do very patient, long-term comparative experiments to understand trophic controls and how important they might be. The interactions are hidden from us with just inspection, and even with diet studies, they're hidden until you kick it and see, or do the sort of work that Jim Estes has done over 30 years in the ocean. Okay, so the absence of fish wipe out that 4th trophic level experimentally. These small predators suppress all of the grazers that matter, and it's a green world. And as some famous ecologist, Harrison Smith-Slobodkin, predicted it would be, it's a nitrogen-fixing world, because nitrogen is the limiting nutrient in the system. So if you're not grazer limited, you're resource limited, and the winners are those nostoc with a nitrogen-fixing heterotoph, a free-living cyanobacteria, and epithemia. You'll hear a lot more about epithemia, but it's a fascinating diatom that has endosymbiotic nitrogen-fixing guests inside. So it has cyanobacteria that live inside it and make the symbiosis a nitrogen fixer too. And that's 90% roughly of the epiphytes towards the end of these experiments and towards the end of our summers in a nitrogen-limited world. So, okay, context dependence. Very strong result. The first year I did these experiments, went out and doubled the number so I could repeat them and tease apart the separate and interactive effects of the fish. And in both years when I repeated these very laborious experiments, there was no fish effect whatsoever, 0. So whether fish were present or absent, the algae collapsed back to detritus within 2 or 3 weeks. This makes a pre-tenure professor extremely insecure. It's very troubling. You know, the unsympathetic, Ernst Williams' unsympathetic law of nature is always initially a very traumatic experience. It's just later that you realize you've learned something neat from it. So what did we learn? We learned when we started opening our eyes, and we as Tim Wooten, who's now at University of Chicago, and myself, we learned that there hadn't been any scouring winter floods. California had entered one of its drought periods, and there had been no scour that winter. And because there was no scour, we got 1 to 2 orders of magnitude higher densities of large instar caddisflies. This guy, Dicosmicus, is one of the world's largest caddisflies. It gets over 1 inch long. And it's long enough that very few predators, only birds in our system, really eat it. It's too big for our fish. So the algae start to bloom as the early spring and late spring come on, but it gets these bald spots nibbled into it by rampaging herds of Dicosmicus that don't care about predators, okay? So here's the experiment Tim and I did to show that that was true. It was simply Dicosmicus presence or absence crossed by steelhead presence absence. And you can see that where Dicosmicus were present, the substrates are grazed clean and no floating mats form. But where they're absent, the diatoms and filamentous greens are overgrowing all of the substrates, and enough is formed to form modest floating mats. Remember, we started these experiments in June on a grazed background. That's why we don't get the huge algal blooms that you get in a normal flood winter, because you start out from a grazed condition and recover it during the warm drought season when conditions for algae are not quite as good because it's getting hot. Here are the data. No Dicosmicus more algae. That's obviously the big story. There's still a steelhead story, and steelhead are still at the 4th trophic level, but they're not the real story anymore. The real story is a 2 trophic level story. So over the years, we've repeated these experiments during 2 more flood years. 97 got another 4-level fish effect. 93 got algal bloom with mats, but then the mats were exported with an unseasonable, very late, early summer flood, and the midges didn't recruit. So that in the absence of these midges, fish were good for algae. So it seemed to be that helped nail down the tuft-weaving midges' importance. So context dependence, here's temporal context. If you have disturbance, you can reset the food web to earlier successional grazers, which certain predators can control and get either a 3 or 4 level chain out of it, a chain that will support predators. If you don't have disturbance, either because it's drought or because we've done disturbance removal experiments throughout much of the arid West, then you tend to have food chains where these players may all be present, but they're not dynamically linked to algae, and they're not feeding very well either. They're not getting a lot of the algal nutrition, so they're not growing well. And the algae is going into defended grazers that grow slowly, so they're slow to recover from disturbance, scour, but once they take over space, they're the top of a 2-level food chain. So theory, when we did these experiments, said that disturbance should shorten food chains, and that was theory done on computers with no temporal context, no spatial context. If theory combines with succession, you can see that at least initially it may release the lower trophic level or whatever is fastest to come back, but eventually it probably will divert more early successional, highly edible stuff up to predators, and in that sense, lengthen food chains. So we do care. One of the regimes in the food web that we care a lot about is whether algal, carbon, nitrogen, energy will go to a short food chain, end up in some trophic cul-de-sac here, maybe be inedible itself, or whether it will feed predators. And we care about this for not only actually obviously academic reasons, but people who like fish and like clear water also care about that. So there is a spatial context that matters too. And any of you who've hiked in watersheds know very well that as you leave the lowland main stems and go up into the tributaries, the substrate gets coarse and bouldery, and you can very soon leave a regime where disturbance happens several times a year with big floods and get into a regime where it might happen just every 10,000 years or every 1,000 years with landslides. So when you get up into these bouldery headwaters, the substrate gets a lot more stable, but of course it gets a lot less productive as the stream gets shaded. So you're going along a disturbance and productivity gradient that's actually partially quite predictable, just given how much drainage area is feeding. A small drainage area in a headwater stream or a large drainage area feeding an entire main stem. So we've been interested in how that spatial context plays out in terms of predictive mapping, because obviously if you go into the headwaters, you've lost the disturbance. So that clue is missing. What about productivity? What's that going to do? Or the other factors that change with that context. And I've been fortunate to start working on this with a team that's provided really good technology tools for advanced mapping and advanced environmental sensing. We've provided our own, ecologists are tracing now a lot with stable isotopes, and my group has done some of that to try to understand the spatial scales that we need to study and how resources flow in and out of consumers, what are resource sheds. But the predictive mapping is a new frontier for all of ecology. It's going to be highly available. How many of you in the room are actually working with LiDAR data now? So it's incredible what you can, well, I'll tell you a little bit about the details of those who aren't familiar with it in a minute, but the density of points you get is just amazing. So given a LiDAR survey of this watershed, which is how we've colored it in. Here's the South Fork Eel River that I just described to you with some terrace meadows, and here's some of the tributaries we study. We're taking this approach. How do the environments change up and downstream as a function of the topography and the stuff that we can infer just from a digital elevation map of the system. How do environments change that constrain species? How do their species' physiological performances or fitnesses change? And because of their performance changes, how do interactions change? How do the outcomes of interactions? For me, that's the bottom line, change as you go up and down drainage networks. So march up and down, do mensurative experiments, and measure where the rules seem to change. Is it a threshold? Is it a gradient? And then once you find some of those changes and have them mapped, why do they change there? What are the controlling environmental variables? Test that by rearranging those in the watershed. Then once you think you understand that, probably think is the operative word here given Ernst Williams' cautions. Once you think you understand that, then get your modeling friends to help you say, okay, I will predict that the domain under this trophic controller, that trophic controller will expand or contract with the changes, the big changes we're all worrying about, climate, biota, land use. So these bits will increase, these bits will contract. How then should the whole basin-wide production of nitrogen or mosquitoes or fish add up? How do you upscale? This is rather difficult. And so I'm going to talk about the modest progress that we're making with this kind of predictive mapping approach. You'll probably be left with a keen appreciation of how much more there is to do, but I hope you find some of this interesting, if only historically. And here's some history in my life. Bill and I were graduate students together at University of Washington. He came down and helped me map my study stream in Panama, and it took us 3 months to map 3 kilometers of the Rio Frijoles with 10-centimeter contour intervals, which matter to fish, and above and below the base flow, so we could see how the habitat expanded and contracted. So I was left with this wonderful bathymetric map. Bill got a fever out of it. It's very hard field work. He is now the head of the National Center for Airborne Laser Mapping, which is a research grade facility that you can piggyback onto your NSF grants if you don't know about it. And they flew over 25 square miles of the eel and shot the LiDAR in just an afternoon. It takes a lot of effort, many, many weeks of effort to program that up into data, but it's much, much quicker than the field work used to be. And for those of you who haven't seen LiDAR data, it's mind-blowing. These are digital data representing what looks like a color photograph, and we actually have much higher resolution than this now. There are maybe 10 to 18 points that fall in every square meter that the plane flies over. I probably should mention that the plane has a laser mounted in it with really high reporting back to an instrument in the plane. That's how it's working. There are water-penetrating LiDARs that aren't eye-safe yet, so they're used offshore, but pretty soon they're going to be eye-safe. We hope to actually get one this summer to do the bathymetry. So from the algorithm that is from the first return from the reflection back to the plane, you can look at the canopy surface of forest. And you can even start to pick out the shapes of large trees, like these Doug fir that have been colored a dark green, or the more globose oaks. So these are just digital data that have been color-coded by height interval to start mapping what's actually a fairly realistic picture of our fairly, in terms of conifers, fairly species-poor system. We have a lot of deciduous trees, but not a lot of angiosperms, but only 2 important conifers. And so then an algorithm that comes from the last return lets you look through the forest at the bare earth. So you can say, why is this forest so lush on a south-facing slope? It should be drought-stressed. Oh, take the forest away digitally. There are a lot of landslides there. The soils are deeper. It's just amazing how much progress you can make, and you can do all sorts of course, the environmental modeling. Of course, for the environmental modeling, you need environmental sensors to get a little bit of reality. And this, I feel, is where the field is going to be slower, because this is hard. More and more watersheds are getting these wireless infrastructures that support dense sensor networks, and the sensors are smart and talking to each other in ways that save energy and deal with small local failures. But it's still really hard work to get these things up. Here's an arborist shooting lines over, so Colin Boda in our group can climb up with 100 pounds of solar panel and radio on his back, and we call this generation of young field people eco-geeks at Berkeley, a person who can climb trees and configure at 110 feet. He's setting up, you have to do that, unfortunately. You have to configure them up there to get things right for the signals. Here's the manager of the Angelo Reserve, the grandfather of the people who gave that land to the Nature Conservancy way back when. So they're both up there in the trees, setting up a wireless system and only complaining a little bit. Redwood height 110 feet. The stuff they are dragging up is hundreds of pounds, arms very sore. Once they get it up in these big trees, though, it's inconspicuous. I really like that as the manager of the Angelo Reserve. Tourists don't see it, visitors won't see it. It is not an eyesore, although it looks like it might be one, and this is what it's doing electronically. It's bouncing signals from our sensors and also our telecommunications, so it's made the place safer to work, among the tall trees and then to a mountain peak where it gets onto a high-speed line in the nearest town and onto the internet. So the dream is that we can all watch sap flow, or somebody in one of our reserves spotted a wolverine on a camera. You could watch your animals, watch your algae grow, as I like, or other things real time on the web. In reality, we're just doing MET stuff on the web now, and this is all still bleeding edge, still it's a work in progress. But I think in the next few years, we'll really get some very useful, at least simple data from it. So that's where we are, and many other groups are at similar phases. Your generation is going to have to deal with the data torrents to interpret these environmental data and make ecological sense of them. And that's not going to be trivial. But let's tell the story now, get back to the story of these ecological regimes, these productivity, disturbance, moisture regimes, and what they mean for algae, and the challenge then of upscaling, another thing that ecologists have to focus on. So I want to have you reflect that here's Cladophora. We figured today, I think Gabe and I figured that it is about the diameter of his hair. It's about 50 to 100 micron diameter filament. So that's, depending on how coarse or fine your hair is, it's human hair size. It branches, though. That's important. And it has all these wonderful nooks and crannies in it, some of which are low oxygen, some are highly oxygenated, some exposed to light, some shadowed. And so you can think of what we're looking at here. Here's 100 micron scale. These diatoms are 20 to 50 microns. These diatoms are, you'll see more of cochineus these are the ones that are going to turn Cladophora yellow and made the algae look so scuzzy in my experiment. They're injuring the host. And this is probably microbial growth that is feeding on carbohydrates that are leaking out of the injured hosts. We see this again and again, and that's our interpretation, at least at this point. These microbes are doing really interesting things biogeochemically. Some are oxidizing iron, some are methylating mercury. We don't have a big mercury problem in this system, but mercury in food webs gets a lot higher as we get more algal productivity. So there are all these interesting biogeochemical ones. The ones I'll focus on are the nitrogen fixation in the diatoms. But there are all these really interesting things that might scale if you understand controls on algal food webs that are mediated trophically and hydrologically. If you understand that, you might understand export of iron, silica, mercury, or nitrogen to the coastal ocean, which is a scale that matters to global change. So just consider we've got a web in a web in a web here, the 3 levels of webbiness, and a lot of possibility, I think, for upscaling. So let's consider the fate of this algae. We've got the Cladophora, we've got the diatoms that grow on it and the bacteria that grow around them. So if that Cladophora grows and dies in place, then it's going to go back to the atmosphere as CO2 if it died in a place with oxygen. It's going to go back as methane and release its nitrogen if it died in an anaerobic place. So that's the simplest fate. And that does happen. But a lot of the Cladophora gets eaten by a bug, but the bug might escape and do what aquatic bugs hope to do, emerge as a winged adult and find a mate. So you've got bug flux out of the river. And some of that bug flux is going to be eaten by lizards, spiders, and bats, and some birds. So this might be the fate of our algal nitrogen carbon. But if it escapes emergence, and if that algae is eaten by a bug, it gets into an aquatic food web if it's eaten by a fish, say. So it could stay local or move around in the freshwater food web. And if it escapes all that, it'll go down through an intertidal food web in the estuary, which is narrow in the eel. And if it escapes the wall of mouths that are waiting for it here, it might get into the deep ocean canyons and get buried. And then it will be out of circulation for a while. The canyons of the eel are very deep and politely collect stratified sediment. So we'll talk about that, I hope, if I can hurry and get to the end of the lecture. Let me, though, just give you some sense of food web controls on algal production changing down that river drainage network. And this is a really simple example, but it yielded some surprising results. Any stream ecologist would look at this river, which this little tributary, dark, shaded, drains 2.6 square kilometers, no algae, no surprise, light limited. Algae don't like the dark. So Camille McNeely looked at that and said, what are all these little rock-cased glossosoma doing there? Those things up here in these headwaters are just a couple millimeters big, so I never noticed them. They look like little sand grains. She has good eyes, and she pulled thousands of these things per square meter out of replicated reaches of this tributary and released on a field scale 10 centimeter turfs of very highly edible diatom, colonial diatoms. So she got that algal release. This is not a place where algae are light limited, it's a place where the grays are limited. It's a 2-level food chain. So these things are doing it, and here's the watershed, just a very small one. So here is another experiment that Camille ran, just easier experiment to do, but place unglazed tiles on the bed where they're exposed to grazers, the black histograms, or lift them. In our case, we now use these little plastic food storage containers that grazers can't climb. So you eliminate most of the heavy cased grazers, and that gives them relief from grazing. That's the white bar. You do that in a 1 kilometer basin. There are grazers there, and the isotopes tell us they're eating pure algae. These glossosoma are made of algae as far up in the drainage as we can collect them. We can't find the algae, they can. They're made of it. But they're not controlling it until you hit a threshold at 2 square kilometers where they do make a big difference in algal biomass, and they continue to do that all the way down to 500 square kilometers. So we've continued those experiments downstream. So that's a regime change in control of algae. So here's the tour. Here's a 0.5 square kilometer basin, 0.56. Caddisflies are eating algae, they're not limiting it. 2.6, where she did the removal experiments. They're eating and limiting algae, and if you take the caddisflies out, you can move the algal isotopic signal into the edible, naked, mobile prey. And if that's true, you should see that algal carbon moving into predators. In her experiments, which lasted just 2 months, we didn't see that. We saw it in the mayflies, but not in the stoneflies or the salamanders or steelhead. So maybe move that downstream, you might see it faster, carry this experiment out longer, maybe. But what we're starting to see here is a threshold where it's a threshold that illustrates the predictive mapping game we're trying to play. That is, if we got a disease like the microsporidium that eliminated glossosoma from Michigan, if we got that microsporidium at the eel, biotic change, then the threshold drainage area from which these predators can start deriving their carbon from algae would move upwards in the drainage network. And with a little energetics calculations, you should be able to compute more steelhead or salamander production and understand what that would mean. So that would link a microsporidium through a bug that they don't eat to salamander production through this network. And with good mapping, you should be able to maybe compute basin-wide, if nothing else changed, how much more salmon might that produce. Okay, here's 17 square kilometers, a bigger tributary where it's half open. There's enough algae there to support both the predator-bearing chain and the armored caddisflies. So they can't eat enough of the algae to eliminate it from this flow now. But the predators still have a terrestrial signal, a terrestrial leaf litter signal isotopically. And then you go to our main stem, and there we see enough production and emergence of bugs that a lot of our predators seem to have the algal signal, although we can't necessarily distinguish algae, which are heavier than leaf litter in the sunny main stem. We can't necessarily distinguish those from predators that are eating very old decomposed carbon in soils. So we can't confidently say at this point that it's an algal signal rather than an old soil signal, because we found heavy carbon in a millipede once, and that discouraged us from making these interpretations. But I want to now turn to the Cladophora story in the main stem. This is where Cladophora starts blooming. This is a real threshold, about 100 square kilometers here. The reason we know as much about algae as we do is thanks to this man, Rex Lau, who's trained a bunch of students that informed me, and actually we've been lucky enough to have him collaborate with us in the last few years. One neat thing about Cladophora, I mentioned it branches, it gets meters long. This is probably an underestimate of how much it increases ecological surface area. And you can see these blooms, while they last, are quite big. So that's a lot of surface area in terms of the Eel River reach. So what's going on that surface? It's getting covered with epiphytes, and this shows up macroscopically in a way that we're very hopeful might help us remote sense it. New Cladophora is green, then it turns yellow in middle age, and rusty red instead of gray and white in old age. And here's what's going on. This is the stuff that turns it yellow. It's that Alka-Seltzer tablet cochineus, and also stalk diatoms. All of these early successional diatoms seem to have to touch the primary host substrate, so they don't get all that thick. And their carotenoids turn it a yellow color, but not a rusty color. But later in succession, those things get overgrown by this character, epithemia. We have 3 species. You see 2 here, small one and a large one. And this is the one that has those endosymbiotic cyanobacteria inside, the same one that took over my experiments in the no-fish treatment. So you see the cyanobacteria here in the living diatom. When this diatom dies and you just have the glass frustule, you can see them more clearly, looking stressed. So this is what's going on when the Cladophora turns rusty. Monolayers. Here's the naked host. These things are 5 to 10 cells deep, piling up on each other's shoulders. So that much carotenoid has a rusty brownish color. So these are, I have to, at this point, thank Paula, who knows the algae very well, a former Rex Lau PhD, now postdoc with me, and Jill Welter, who is my postdoc now teaching at a small college in Minneapolis, and a bunch of other engineers and friends who are studying algal-based food webs, some of them more on the bug flux and fish side, some of them more on the algal microbial side. Here is Paula and Jill's work. Paula doing the laborious counts of biovolumes to get the x-axis, and Jill figuring out how to keep a gas chromatograph alive in the field and getting the nitrogen fixation rates. These are our first data. We hope to get a lot more of these, but you can see that the green stuff is not fixing very much, but a little. Yellow stuff fixes a surprising amount. I'm actually puzzled by why that number is double, because there are some free-living fixers on it, but I didn't think that many. But here's where fixation increases 5 to 6-fold with epithemia. So more nitrogen coming into the river now with more epithemia biovolume. And more, well, before saying that, it may make a water chemistry difference. Here is another threshold in our map, log scale here, but look at the jump up in dissolved nitrogen at 100 square kilometers, where it's light enough for these fixers and other fixers like alders to start kicking in, and free-living cyanobacteria. We think these are really important. Our alders, the measurements we've done so far suggest they're not leaking very much nitrogen back, at least during the summer season. So here's late summer when the epithemia is really cooking, and you can see even more nitrogen at that drainage area increase. When a blue-green fixes nitrogen, it could turn it into a toxin or a protein, a yummy protein, and these guys are yummy, as I was saying at our grad lunch. Here's the midge that I mentioned before. Its gut is full of diatoms, and the diatoms are all empty, and they're all epithemia here in this typical representative gut content sample. And the midges actually clean the rusty red diatoms off near their silken retreats. So you can see them turning the rusty matrix algae back into green as they groom it. So as they are eating this stuff, they're one of the larger and more common large insects to emerge from the system. We use these emergence traps. And here it is coming from a slimy algae that doesn't get epiphytes, or new Cladophora or middle-aged Cladophora. And here you can see the real bug biomass is coming out of the rusty algae. That's an increase of about 25-fold in insect biomass emergence with rusty stuff. So these are just data that show that. And I'll skip to a picture which is not taken at the eel, but in the near the Consumnes drainage basin, where we've also worked, just to get your attention about the importance of bug flux And this little cloud of smoke that Bill Rainey of my bat collaborators photographed is coming from the same bridge tracking that bug flux. These are bats. So these are thousands of Mexican free-tailed bats that each of which eats its body weight every night in mosquitoes, or mosquitoes and other emerging insects, in-flying insects. So this is an interaction we really need to care about as California warms up and water distribution changes. So I'm actually going to move through this really quickly because I want to take you to the ocean. We are interested in how bats track bug flux through a complex drainage area. The rules seem to change. I apologize for showing you this so quickly, but when you get into the noisy tributaries, the bats stop surface skimming, which is a highly efficient way to trap emerging insects, and they start picking perched larger insects off of foliage at night, behaving like warblers. Different species of bats do this. Why do they do it? Probably has something to do with the acoustic environment of a steep, noisy, burbling brook versus a quiet lowland pool. So predictive mapping, this summer we plan to record that sound, see if we can drive bats away from quiet pools with riffle noises, and if so, have some prediction for saying, okay, so if we lose most of our discharge, the areas of noisy, turbulent habitat will increase, the areas of quiet pools will decrease, and you'll gain this much area where emerging insects will be out of control of surface skimming bats. So grape grower, if you take a third of my water, this is a possible consequence. This is an issue in California. So that's another predictive mapping scenario that is still just kind of a pipe dream at this point, but I think with the manipulation we'll be closer. Okay, so I've gotten you through this stuff, not too much of that except in the disturbance talk. But now let's talk really quickly about the marine. The eel's interesting to people who watch rivers and oceans from space because here's the Bay Area with its massive chlorophyll bloom that's derived not only from every toilet flush in my community here, but also from the entire Sacramento and San Joaquin agricultural valley. So that's big, but why is this one so big? This is the bloom from the Eel River. And the Eel is a much smaller basin, about roughly 10,000 square kilometers, 9,700 square kilometers. And it has very light land use. It was brutally logged. There's a lot of damage there from the logging practices of the 50s through the 70s. But there's now just light dairy, a little bit light agriculture, not dense settlement in this whole Eel Basin. So what's going on that's feeding phytoplankton blooms? Of course, part of it's ocean upwelling, but some of these chlorophyll maxima are out of sync with the strongest upwelling and more in sync with river runoff. So there's something the river's providing that's interesting. And I'll rush through these. These are just data summarized from doing lots of cross-stream transects of where are the algae. You can see here drainage areas getting towards the main stem rivers here. Early summer, the rusty and yellow and green cladophyra are starting to show up after you get larger than 100 square kilometers. They show up more later in the summer. Here are some of the free-living taxa that are also fixing nitrogen, but not as fast. So we've expanded. This is our little postage stamp of an 8,000-acre reserve that we know rather well. But the smallest graduate student in my group is taking on the largest problem. She's doing these transects all the way down the river to the mouth. And Charlene is lucky because you can wade the eel. It gets wider, but stays shallow. You can even wade the estuary at low tide. So this is a wadable river, and it's a steep river, so it stays gravel-bedded all the way down. So these algal phenology rules seem to apply. You get the same kind of phenology here as here. So we're very lucky in this system in terms of our ambitions towards upscaling. Just a picture of the shallower, wider basin where a lot of her sites are. And too little time to talk about this, but because we have all of the drainage areas and we have figures of how many free-living and epithemia fixers are there at various drainage areas and how much drainage area is there in the basin for that interval, then we can do a nitrogen loading budget. And I get a number, 2,400 kilograms per year from the whole basin loaded into the ocean. Now that's not a number that can compete with ocean upwelling, but ocean upwelling is weak and not happening so much in the late summer and in the winter. So it's possible that could make a difference. It's also possible that the chlorophyll is benefiting from something else the river is kicking out, and that's still, we have a lot to learn there. But The optimism that we can study this photogrammetrically is kind of shown here. As we get downstream, there are bridges. You can see rusty yellow and green cladophora mats from the bridges. So not a lot of bridges, but some. And of course, we're trying cameras in trees and cameras in toy helicopters and that sort of thing too. Charlene's looking at the wall of mouths that is the last thing these algae have to go through before they make it to the ocean. And there's quite a wall there. She prepares local seaweeds, the ulva, which is supposed to be the most edible marine alga, and our even yummier river algae, and puts them out in habitats like this, where the gravels, this is the estuary mouth that's still got a lot of gravel in it. That gravel is infested with 2 species of isopod, and 2 species of amphipod, and 1 isopod. And so she's come up with this extremely clever, simple way to split a disposable pipette, mount the algae, and put it out in cafeteria trials for these beasts to work on. And the amphipods love the rusty stuff and devour it immediately after they find it, and like it a lot better than ulva, which is supposed to be the lettuce of the ocean, but is obviously inferior to our river algae, at least in their mind. So there is a wall of mouths. Here we are trying to understand something more about the controls and how this stuff gets into the ocean food web, but a lot of the algae makes it past all of these fates and gets into the deep ocean. Now, we don't really know what fraction. We could maybe think about that. But there are cores. One of the reasons the eel has also been really neat to study is that a group led by Chuck Nitrower at University of Washington has been funded by the Navy to do very detailed stratigraphy of cores that are in the deep canyons. At low sea stand, the eel carved deep canyons like Monterey Bay Canyon. And these things then trap 30 to 50% of each year's export of sediment that the eel kicks out. And it traps it in the deep canyons in a very polite fashion that record annual varves. So when I heard about this, I asked Chuck if anybody had looked at diatoms in those varves, and they've done everything else, carbon and sediment analyses, but nobody had looked at diatoms. So Jack Scully is doing that, and he's actually got pay dirt. This is my last example of why you should welcome Ernst Williams' unsympathetic laws of nature. So when I was trying to correlate records of discharge to the magnitude of algal blooms, I really just wanted it to be simple, please, low, if you get a sub-scour year, this scour is kind of a threshold phenomena in a gravel-bedded river, and 120 cubic meters per second is our threshold for the whole bed unlocks and goes. So low flood year, drought year, low algal bloom because the armored grazers haven't been wiped out. And then should be big algae years and high flood years. So what's that point doing? Why, distressing noise? Well, it's not distressing anymore. This noise has really helped us with the marine interpretation in an exciting way. This is the year that a late flood exported the algae after it had started to grow, and it just didn't regrow very much. So we get that kind of noise and other embarrassing to me, with my simple story, signals. But this has allowed Jack to tease apart in his cores real productivity, the real magnitude of algae from just discharge that might have kicked up and export stored algae. We don't know too much about how much a year's worth of algae is stored in the deep pools in the rivers. Is it stored a year? Is it stored 5 years? Does it take a 10-year flood to get it out? Well, it turns out it looks like it goes every year because it's a short record, 18 years, but those 18 years, the correlation of freshwater diatoms here is stronger with my actual algal surveys than it is with discharge. So that's very encouraging and exciting that the epithemia in particular, which has a robust frustral and is diagnostic of big cladophora blooms, might be a really interesting paleoproductivity indicator. So the oceanographers are excited enough by this. This is very preliminary analysis, and Jack's still working this up and has just barely finished the counts. But Chuck Nitrower is thinking maybe if this holds up over these 18 years, we could use the diatoms to go back and resolve some of our less certain dates earlier in the record. So what could be looked at in terms of climate effects on integrating river and nearshore processes are 100 years of annual resolution in the deep canyons, and then up on the shelf they can get thousands of years, but not with annual resolution because there's more bioturbation, but maybe with 3 to 5 year resolution, which would be good enough for PDOs or other multi-year phenomena. Okay, so really quick 5-step plan for map-based prediction. document thresholds. I've shown you a 2 square kilometer drainage threshold where grazers limit algae. I should say I'm doing this all in drainage area. It doesn't have to be drainage area. It could be any other aspect that you can look at from a map. But use these maps, these incredible mapping tools we have to make hypotheses about how nature should be in the field, then go to the field, find your unsympathetic nature has refuted your firm and vivid prediction, and figure out why you're wrong, but with a very alerted mind. So 10 square kilometers, predators can get fueled by algae. 100 square kilometers, all sorts of things start with the blooms of Cladophora, things of biogeochemical interest. You rearrange the controls, and here's the white stuff we haven't really done enough of yet. Validate predictions in new sites. They can be, you may have the same drainage area but less forest, so you should be able to make a directional prediction if you think you understand how productivity is affecting this. So I was talking with many of you today about the deficit of prediction or forecasting in ecology. I think these map-based tools are something ecologists, in fact, are shy about making predictions. And the map-based tools should really give us a lot more confidence that we can at least make directional predictions. So how will changes in climate, land use, or biology change the areas that are under a given kind of regime by changing light, by changing disturbance, or by changing the rules in the food web? Then the hard part, basin-wide scaling, I used to think that would not be approachable in my lifetime. I'm getting a little bit more optimistic now as I work with earth scientists who can constrain basin-wide outputs by paleo samples, or folks that can model and think about upscaling. So a wrong prediction is a very useful prediction because you can go and see why you're wrong and what perhaps larger scale death trap for salmon or storage elements you're missing. Okay, we all care about the future, and if you invest in a place, you care about its future. So this is the very evocative Sheffer diagram of a condition changing maybe from a state we like to a state we don't like as the environment changes. The big stressors in these western basins are, will the landscape fall apart with more rain as that unleashes landslides? Or will we lose water? And if we lose water, what roles are trees playing in maintaining water? There's a huge amount to do. I'm optimistic that these technology tools will help us become a lot more useful in forecasting if we can tame the data torrent, get the systems working, visualize them, share them. It's a huge amount of work for your generation, the young generation, to accomplish. And I always fall back on thinking of myself as Homer. As I left, the kick it and see experiments that you do over tens of square meters as an experimental food web ecologist in one kind of environment, and you take on spatial and temporal context and work with earth scientists and atmospheric sciences, you enter a world that's challengingly multidimensional. Just as Homer, when he was getting away from his sisters-in-law, he walked through that strange wall behind his couch and ended up in a 3D world on the special. So here's Homer looking scared, and there should be muddy boots on that person. Food web ecologists like myself are scared of this. But I have to thank these people who are adding to my knowledge of these dimensions. We food web ecologists aren't scared of bears, and this guy isn't scared of big global thinking, and this person isn't scared of math and as Feng, and he's not scared of technology. Mickey Hanzo, my engineering colleague who uses delicate instruments in the river, and these guys know and love and share their love with algae, and she and Jill and Jacques, are very good biogeochemists, showing how tracers can reveal spatial dimensions in these webs and also what they might mean as they scale up. So this is where we do have to be very collaborative and open in our thinking with other disciplines, and there's a whole world out there to understand a lot better if we can pull this off. So it's in your hands largely. So thanks.
along the lines of what my colleagues and I are trying to do in Kenyan savannas, only you guys are doing it much, much better. And I guess my question is, so the wrestling with this very similar sorts of issues, and one is that it's, within a food web, you can manipulate the system, and as you said earlier, that's very important for elucidating mechanisms, hidden mechanisms that you just literally would never be able to see otherwise, no matter how many replicated systems you have. And obviously, unless we start collaborating with the mining industry in totally new ways, we cannot manipulate topography or
right.
So do you expect the same kind of hidden mechanisms that we get in food web studies when we're looking at what are essentially very data intensive but still correlative kinds of relationships like in your watershed?
That's a good question, but because they're not dealing with idiosyncratic biology and with the behavior and all the complexity we have, I think they can do better detective work on systems. So, in our happy 35-year marriage, Bill and I have always had a tension, which is he can go to a landscape and learn a lot with a touristic experience of just, seeing it for a few days. I can't understand anything unless I've lived with the system for 3 months, and you know exactly what I'm talking about. Fantastic work in Kenya that you guys are doing. I think that they have a lot of modeling tools that ecologists are starting to pick up and use effectively, but you only have in models what you put into them. And the surprises in our food webs are, we need to keep working it from both ends, obviously, to get the right components in the models. I guess one place I think earth sciences has been stronger than ecology is their respect for observation of process. So ecologists tend to not think so hard about which site will I make my measurement in. In fact, that's sometimes considered bias. You should maybe stratify a little bit, but then take a kind of blind man's bluff approach so you don't bias yourself in your data collection. The geomorphologists go, where's the process clearest, and let's document the rates, and then understand why this was a good site to study, but you chose a bad site over here, so you'll never see what's going on. So they're looking more for model systems in nature. That's powerful for them. So it is an interesting, I think all of, not only will we get all the bits of information to add together, but we'll get new tools to think about nature, even to apply to our own fields. Does that kind of get at your question?
Yeah.
Yeah. Okay. Good. Yes.
You spoke about lateral flows in the context of connecting, obviously, the aquatic with the marine. But I was curious whether you think, for certain aspects of thinking about the ecology of streams, you actually have to think about the lateral flows, between riffles and pools on a much smaller scale of kilometers, and in particular whether that is driven by fluxes of nutrients, but to what extent it also involves the decomposer community.
Yeah, Jock Finley, for his PhD in the eel, did really nice work that opened my eyes to that. And the carbon isotope in streams had been very controversial because sometimes it looked like it was terrestrial, -28, and sometimes it looked heavier, and sometimes it looked lighter, and nobody had figured out why until Jacques put it together, I think borrowing from discoveries in the lake literature, that if algae are carbon limited, they'll take anything and they'll get heavy because they prefer, quote unquote, the C12 enzyme. But it turns out that if you have a productive river with algae and riffles and algae and pools, then the algae and riffles get a very light signature because they're not carbon limited. They're competing with each other, and there might be as much algae there, very productive, but the flux is enough to deliver carbon. So they stay light, and in the stagnant pools, they get very heavy in C13. So then what that opened my eyes up to, Jacques has always been interested in the scales over which these longitudinal linkages occur. But I won't talk about decomposers right now, but imagine you have insect fauna on a boulder that's just downstream from both riffles and pools. It's in a fast-flowing riffle, and you've got scrapers clinging to that rock and filterers. And you open them up, and the mystery matter in their guts looks identical, and it actually is taxonomically identical. If you look microscopically, you can see frustrals, but they're the same frustrals. So if you do Jacques isotopes on them, you can see that the filters are feeding from the shallow pools, and the scrapers, as you'd expect, are feeding from that local riffle. And that leads to predictive mapping. If these guys are competing for space, but they're eating from different photon sheds, then if you shade the riffle, the filters should start winning. And if you shade the pools, or if you change management that changes the area of pools or riffles, then the pool fauna, pool feeding filters should start winning. And you could start by understanding explicitly the source areas, the environmental source areas that are coming from different parts of the environment, feeding organisms that are interacting in the same web. We can start to do what has been a big challenge in food web ecology, is linking diet to dynamics. Why so often do you have the population regulating pathways take a different route through the food web than the energy flow pathways? So if someone's most important predator or most important prey, they may be irrelevant to their population dynamics, or not very relevant. So this shows how the environment might link to that in changing interaction strength. So those scales are of extreme interest to us. It goes the other way too. You can have interactions that block environmental fluxes of resources. So these we call resource sheds and resource shadows. So if you have those, then it's the interaction that changes the environmental flow. So those things are all upstream, downstream, sometimes cross-valley, and vertical linkages that clever detective work with isotopes is revealing in a number of systems.
Why don't we thank Mary again, and there's a reception just outside.